Hello everyone, welcome to Pharmaizer. I post videos on pharmaceutical topics explained in a very simple and easy way. If you'll find my video useful for your studies, don't forget to like and share my video and subscribe to my channel. So today we will discuss on lab diagnostic system. We'll discuss about the introduction to diagnostic system and types, then about the lab diagnostic system, its advantages, disadvantages, and how computer helps in various laboratory testing. One more thing, this will be the English explanation video. For if you want to watch the Hindi explanation, for that I will add the link in the description box, which is already been posted. So if you want to just understand in Hindi, you can just watch that part, and if you want to just know, or you don't want to understand it in English, you can just watch this video. So anyways, let's talk about what is diagnostic and what are the various types of diagnostic test. So first of all, diagnostic means what? Diagnostic means to detect, to identify. In health or medical field, diagnostic means identification or detection of the diseases. And how it can be identified? By various examinations of its various signs and symptoms. Simply, we just observe various signs and symptoms. According to that, we collect various samples, or just we produce images of various body parts, or simply just we detect the symptoms, and according to that we try to identify the various disease which can occur. So that is basically diagnostic system. So here the various types of diagnostic is, first of all, clinical diagnostic system, which is a basic type of diagnostic test for diseases, where the doctor or the physician just observes various signs and symptoms in a patient's body and tries to identify the disease. For example, if any person is having just a normal fever, so he will have chills in his body, and then he will be his temperature will be high, heartbeating rate will be little fast, maybe he will have some cold and cough also. So that is normal viral fever. So by observing the just symptoms and signs, any doctor can just tell that he is having normal viral fever, and he will just give the medicine. So there is no requirement of any complicated diagnostic tests. So that is simply clinical diagnosis. Now comes the laboratory diagnosis, where we generate the laboratory test results by testing on various samples. We take the samples from the patient, and then we perform various tests on the samples to identify particular disease. For example, to know if a patient is diabetic or not, we just do the urine test, or we can also conduct the blood test to identify the sugar level or glucose level in blood or in urine. So that will be done by testing urine or blood sample of the patient. That will be a lab diagnostic system. So if any disease which can't be identified by its symptom, which can't be identified simply by its symptom, then we can go for sample testing. That is the laboratory diagnostic system. Then comes radiological diagnostic. So radiological diagnostic means where medical images are being taken. For example, if a patient has a fracture in his leg, so for that, in a leg bone, if there is fracture, so a doctor will suggest for diagnosis, because you can say the X-ray diagnosis, because here you can't see just the symptoms and tell that where is the actual proper breakage, how much breakage is there, fracture is there actually, and there is no sample detection test for fracture testing. So there is only one type of procedure, that is to observe the image of the bone to understand how much fracture has been there in the leg bone. So now, and the fourth one is the computer aided diagnostic system, which is an advanced form of this radiological diagnosis, where we just we just go through the images, but not manually, by using computer software system. That is, there are various sophisticated computer software systems which diagnosis which does the diagnosis test, that is which studies the various radiological images. Like for example, we just go for a x-ray. From there we will get an image. The doctor will observe the image and he will say that yes, here is the fracture and all, or there is a tumor. Also for cancer also we can just go for various radiological identification test or diagnosis. So in computer diagnosis, the computer will be used, that is there will be various sophisticated software system which will thoroughly read or thoroughly scan the radiological images of the particular patient, and then it will identify the abnormal areas for either tumor or for any fracture and all. So here the radiological system is you can say is combined with the computer software system. So here the benefit will be that the patient's particular abnormal area will be identified more better way in comparison to manual identification by the physician, because there may be chances that physician will miss any area, but a computer software system will not miss any minimal abnormal area also, and it will, the computer will actually diagnose and find out all the spots or abnormal areas in any particular body of which the scan has been performed. For the body part where the scanning has been performed, in that particular area any of the abnormal areas or any of the abnormal cells growth or fractures can be identified without any error by the computer software system. So anyways, we will talk about computer aided diagnostic system in the next video. We will talk today about the lab diagnostic system. So basically, what is lab diagnostic system? As I just told, doing diagnostic test on various samples of the patient to identify the disease is known as lab diagnostic system. So it is also known as in vitro diagnostic system, because in vitro means laboratory testings, which makes possible to identify the microorganisms causing an infectious disease and to appropriate treatment and to provide appropriate treatment. So in case of any disease which has been caused by microorganisms basically, or if there is any abnormal constraints, chemical constraints in our body, then the better diagnostic test will be lab diagnostic test. So for for any microorganism infection or any abnormal chemical growth, you will not go for x-ray or CT scan or any radiological scanning. You will just simply test your samples and get the result of the abnormality or any microorganism infections. So it is best for the infection causing diseases. They also makes it possible to detect non-infectious disease. As I told, non-infectious, what is infectious? That is caused by microorganisms, and the infectious are those which are due to our own abnormal chemical constant, biochemical constant, that is the various chemical constituents which is present in our body. When it gets abnormal, its quantity actually increases or decreases from the normal value, that will be the non-infectious disease. For example, diabetes, then rheumatoid arthritis, or various urea related diseases or creatinine related diseases, etc. The most basic parameter that is to be established regarding any clinical test are that it should not demonstrate sufficient degree of reliability and validity. So basically, any test which we perform, we just expect that it should give reliability. Reliability means that the result should be correct, and here the computer can help you in getting a very correct result. That is, in lab also you can utilize computer will help to identify the abnormal constituents or the microorganism present in the sample. How that will also I will tell at the end. So basically, if we are getting these two parameters fulfilled, that is reliability and validity. Validity means the result is valid, you can just prove your result to be correct, that is validating. And reliability means reliability is also a type of correct result, getting correct result, reproducible result. That is, if you go for test again and again, you will get the same result, that will be a reliable result. And valid means when you are getting the same result every time, that is, it is reliable type of result, then you can just state your result to be a valid result, that is not a false, that is a stated and proved result. So if these two parameters are not met, then the test value of by assistant by a physician or clinician to arrive at a diagnostic conclusion and form a treatment plan or monitor a patient's progress is waste. Obviously, if you go for any test and if you are not getting a reliable and valid result of the test, then it is of no use, it is a waste, and a physician cannot come to a conclusion that what type of treatment he can provide for the particular disease to the patient. So here computer can help. How? As I told, I will tell. Let's talk about the advantages. First of all, like clinicians rapidly obtain the right result with the appropriate test for the doubtful health condition. So by using computer, you will get the right result, and for which you can just come to the conclusion at what type of treatment you can provide for that particular health condition. Then weak points in the test process can be recognized easily. Necessary changes, if required, can be done immediately. Like for example, if there is means if a physician is getting wrong about any disease, like first of all, he will go for a clinical test, that is, he will identify the symptoms and he will guess that maybe it can be a disease A. But during lab diagnostic test, you found that the test for A disease is negative. Then you can just change to another type of diagnostic, that is, you will go for various other types of disease which can be related to that type of symptom, like disease B or C. So by using computer, you can just simply identify and simply get the correct positive or negative result, and if it gives a fast result. So if we get negative result for A, we can quickly test our sample for B or C disease. Now human errors are minimized. Obviously, if you are doing the laboratory test manually, then there are chances that uh, you will perform, or you can actually, you will not, you can perform some type of mistake during the diagnostic procedure, and there may be a false positive or false negative result. So that can be minimized by using a computer technology along with your laboratory testing. So it is not laboratory diagnostic system, it is different thing. The lab it is not computer aided diagnostic system, my mistake. Computer aided diagnostic system is different thing. It is included with your radiological diagnostic for image identification, image studying, various image of the body part is studied in the computer aided diagnostic. In lab diagnostic, computers helps to test the samples, not creating the images of the body parts. So here the role of computer is, along with laboratory testing, computer aided diagnostic system is included with radiological diagnostic system. So that is computer aided for radiological testing, and here computer is for simply testing the samples in the laboratories. So human error will be minimized. Then diagnostic decisions are very transparent and accessible to all involved in patient care. So the diagnostic results which we get from this computer systems, that is very transparent, means very clear, and anyone can just understand what the result is, what is the outcome, because here the computer is showing you digital result. So that is readable and that is easily understandable by everyone who is who is related to the patient care. So now important to detect any outbreak if suspended. So generally there are cases when we go for testing of any sample, of any patient sample, and for example we are testing for disease A. There may be chances that during testing of disease A we have just found that the particular patient sample also contains some symptoms of disease X, for example disease X. So the computer, because in manually you just do one type of test, but when you are using the computer software system, for example you are testing the sample through computer software system, which will show you, the software will show you any abnormal constraint, any categories of abnormal constraint in the sample. So there may be chances that the computer software system will show you all the abnormalities in that sample. If the software is like that, that is, if the software can identify any type of abnormal constraint in the sample. So during that you may find that you have identified, the computer actually has identified some unique abnormalities which you have not expected in the sample, that is for particular disease X. So by this you can detect any outbreak also. So this is also a benefit. Now talking about the disadvantages. Disadvantages is that not applicable among patients with complex disease. If any patient is having various types of disease, lab diagnostic will not be beneficial either by computer or manually if there is various types of diseases in any patient. Then lack of flexibility and individuality, that is lack in the changes in the method of test depending on the individual patient. So you can't just make your diagnostic test individual, that is according to person you can't change your diagnostic. For example there are some numbers of tests mentioned which is to be performed for detecting any abnormalities, like urinometer is for testing the surface or the density of the urine actually. So you can't go for any other method. So basically it is not, the diagnostic systems are not for an individual, that is common types of tests, they are the common types of tests. You can't make your test individual for any particular patient, so there is lack of individuality and flexibility. But in comparison to that, when you go for radiological testings or computer aided radiological diagnostic test, there you can just identify the particular patient's body and the abnormalities or any type of changes in that particular area or that particular part can be identified, which is based on the individual patient. So that is not common type of identification for all type of patients, that is just an individual type of test. So there is individuality, but in lab diagnostic system there is no individuality, the test is common for each and every one. If you are testing, if you are going through any particular type of test procedure, that particular procedure is for each and every one, each and every patient who are identified or who are being tested, whose sample is being tested for particular type of you can say identification of the sample. So that is a common one, there is no individuality. And then only suitable for patients with a clear symptoms. You can also go for lab diagnostic system only if there is any clear symptoms, if there is no particular symptoms for any patient, like in case of generation of any tumor inside in your body. So there will be no symptoms for tumor, it will grow gradually. For that only one thing can be done, that is radiological diagnosis. No sample can identify your tumor even by lab diagnostic. So then one more, that is your imaging is not included. So in diagnostic system, images are not there provided. You just get the result based on various chemical test which is performed on your samples. What is lab diagnostic test? They are simply a chemical reaction. Like some chemicals will react with the bilirubin present in your sample, or some chemical will react with the urea present in your sample, like that. So it can't uh, give you any image that yes, in the sample these are the red spots which are showing the bilirubin or anything like that. So you just get the chemical result, the reaction after the chemical reaction, either positive or negative. That's that much. Yes, if you are taking a computer system, or if there is any calculation system based with your diagnostic system, just one thing can be done, that is there can be a value. The result can be identified in a value terms, that is you can get a numerical value, but there is no image for your sample identification, just chemical reactions. So these are the disadvantages. Now, how computer helps in lab diagnostics? So basically, just for example, if you are taking the blood sample or urine sample, you are analyzing the result, and this is the main part when you are performing lab diagnostic system, and the computer do much of your work. How? So the sophisticated computer technology can quickly determine whether the level of protein or amino acid or sugar or any elements, like I told, bilirubin, urea, or any such types of creatinine, etc., any such types of biochemicals are within the abnormal range or not. So these same computers can be used to generate results also. So this is helpful in creating the datas also, that is you don't have to print out the result by typing and all. The computer system, the computer software system for lab diagnostic, they will just identify the sample also, they will give you the numerical value also, that the range of the samples of the particular chemical present in your sample, whether it is in normal range, low or high range, and also it will create a report and result, a printable result also you can get. So, and which can also, if the computer software system is more advanced, you can simply, like sending mail, you can just send the, you can just mail, or you can just forward your result, the pathological result, to the physician's software or physician's mail. So that depends on the particular type of software, which type of software you are using. So basically, for example, like urine testings or in blood testing, generally the computer software system, actually it is not just a computer, there are many types of digital, you can say, machines are connected with your computer software system. So the digital machines perform the chemical test within that machine only. So for that, what you have to do, like you are, for example, you are taking sample A and mixing with chemical B, and your result will be obtained as C result. So here what can computer do is, the digital machine will take your sample A, after that you will load your chemical B, the machine will perform the reaction within that machine, the reaction will be performed between A and B, and then the amount of C product which is performed on the basis of the chemical present in sample A and reaction with the chemical B, that is the you can say the particular reagent which can identify the chemical present in the sample or the biomolecule present in the sample. So for that, reaction between A and B, that is the sample's chemical constituent, the sample's biomolecule will give the particular amount of product C based on the amount of sample's chemical constituent. That is, for example, you are testing blood, you are testing urea for urea you are testing your blood sample. So the reaction which will be performed between the urea and your chemical B, that will depend on how much urea is present. The more amount of urea is present, the more amount of product C will be formed with reaction of the A sample and B chemical reagent. So the amount of product which is being created or which is produced, that value will be digitally shown in your computer setup, and that can be printable or that can be mailed easily to any physician, any related physician, or you can just simply take out the print of the diagnostic result and you can just hold it hand it over to the physician or to the patient. So that's all for today. Next we will discuss about the computer aided diagnostic system. So for more videos you can go and check my channel. Hope that you have found my video useful. If yes, then press the bell icon to get the notifications. Thanks for watching and bye bye.